Assalamu alaikum. In this video, we are going to talk about the T cells, what are the functions that are performed by the T cells, and the types of the T cells based on the surface molecules and also based on their function. In our previous video, we talked about the introduction of the T cells, that the T cells were coming from the hematopoietic stem cells. The hematopoietic stem cells were differentiated into the lymphoid series, and then the lymphoid series was further differentiating into the T lymphocytes. So let's talk about the functions of the T cells. T cells perform several important functions, but we can divide those functions broadly into two categories. First is the regulatory function of the T cells. Second is the effector function of the T cells. The regulatory functions are mediated primarily by the helper T cells, which are also known as the CD4 positive T cells, and they perform these functions by producing several kinds of interleukins. They produce the interleukin 4 and interleukin 5. Interleukin 4 is the B cell growth factor and interleukin 5 is the B cell differentiation factor, and both of these interleukins are involved with the activation of the T cell, their growth, and differentiating those B cells into antibody producing plasma cells. Another interleukin that is very important and it is produced by the helper T cell is called as interleukin 2, and this interleukin 2 activates both CD4 and CD8 positive T cells, and then the CD4 positive T cells perform their helper function and the CD8 positive T cells perform their other function. We will talk about it later. The third interferon that is produced by T cell for its regulatory function is the gamma interferon, and this gamma interferon activates the macrophages, and it is the main mediator of the delayed hypersensitivity against the intracellular organisms, and the most common example of this is Mycobacterium tuberculosis. There is also another type of T cells is described, which is called as the suppressor T cell, and the suppressor T cells are postulated to down-regulate the immune response, but the evidence to support the existence of suppressor T cells is lacking. Now let's talk about the effector function. The effector functions are carried out primarily by the cytotoxic T cells, which are also called as the CD8 positive T cells, and these cells kill the virus infected cells. These cells kill the tumor cells and allografts. Within the thymus, in the outer cortical epithelial cells, which are also called the nerve cells, the T cells progenitor cells differentiate into the T cell subpopulation, and this differentiation is mediated by or influenced by the thymic hormones, and these hormones include the thymosin and thymopoietin. The T cells are characterized by the surface molecules that they carry, and these include some of the glycoproteins, which include the CD3, CD4 and CD8 molecules on their surface. The CD4 and CD8 molecules are transmembrane glycoproteins that consist of a single polypeptide, and they may be involved with the signaling via tyrosine kinase. The CD3 molecule, which is present on both CD4 and CD8 positive T cells. CD4 glycoprotein is only present on the helper T cells, and the CD8 protein is only present on the cytotoxic T cells, but the CD3 protein is present on both the CD4 and CD8 cells, and this CD3 protein is made up of 5 transmembrane proteins, and these are also involved in the signal transduction. One of the transmembrane protein of the CD3 is called the zeta chain, and it is linked to a tyrosine kinase which is involved with signal transduction. We will talk about the signal transduction under the topic of activation of the T cells. The T cells are subdivided into two major categories based on whether they are CD4 or CD8 positive T cells. Mature T cells either have CD4 or CD8 proteins, but not both. We talked about the function of the T cells. They perform the regulatory function and the effector functions. These regulatory and effector functions are performed by different cells. Two subpopulations that are very important. These subpopulation come under the category of the helper T cells. So the first population is called as the Th1 cells, and these Th1 cells help the delayed hypersensitivity response by producing primarily interleukin 2 and gamma interferon. So the Th1 cells are involved in the delayed hypersensitivity response. The Th2 cells perform the B cell helper function by producing primarily interleukin 4 and interleukin 5. So the Th1 cell is involved in the delayed hypersensitivity response, and the Th2 cell is involved in the activation and growth of the B cells. One important regulator of the balance between the Th1 cell and the Th2 cell is interleukin 12, which is produced by the macrophages. Interleukin 12 increases the number of Th1 cells, thereby enhancing the host defenses against organisms that are controlled by delayed hypersensitivity response. Another important regulator is the gamma interferon. Gamma interferon inhibits the production of the Th2 cells. CD4 cells make up about 65% of the peripheral T cells, and they predominate in the thymic medulla, tonsils, and the blood. Now let's talk about the CD8 lymphocytes. CD8 lymphocytes perform the cytotoxic function. That is that they kill the virus infected cells. They also kill tumor cells and allografts. They kill by either of the two mechanisms. There are two mechanisms by which the CD8 positive cells they kill the virus infected cells. First is the production of perforins. Perforins are the proteins that are produced by the cytotoxic T cells, and they attach to the cells and they form pores in the cell membrane. Through these pores, the inner content of the cell leaks out into the surrounding and the cell dies. This is the first mechanism by which the cytotoxic T cell perform their cytotoxic function. Second is the induction of the programmed cell death, otherwise called as apoptosis. So these are the two mechanisms by which the CD8 positive T cells perform their function. CD8 cells predominate in the human bone marrow and the gut lymphoid tissues, which was called as the GALT, and they constitute about 35% of the peripheral T cells. In the next video, we will talk about the activation of T cells. Allah Hafiz.